So the real question is, though, how is this going to work in humans? That's where everything else failed, right? Going from preclinical to clinical. So that's the real question, and granted, that's why we're here today, and that's why we're running a clinical trial. I was a project leader. Art Kellerman was the PI on this trial, the initial first-in-human study coming out of Don's lab into the clinic. It was conducted at Grady Memorial Hospital, which was, you saw the emergency department entrance there. It was a phase 2 study. We were looking at safety predominantly. 4 patients, 4 subjects to 1 got drug versus a placebo. It was double-blinded trial. We also did moderate to severe in that trial. And we enrolled 100 patients, and that was 2001, I believe, to 2005. So the primary outcome was safety, and the good news is that we didn't see any adverse effects. Predictably, we didn't think we would see very many because it's a relatively well-known drug. We were looking for DVTs and PEs and some of the other coagulopathic things that you would think of, but we really didn't see anything in this study, and so we felt very good about that. One of the things we did see, however, was a dramatic drop in death in one of the groups. And we went in and we were shown a piece of paper that had adverse events A and B. And one had a 13% mortality and the other had 30. And I went, uh-oh, which one is ours? And then they laid out and revealed the treatment group was the progesterone group. So at that time, I mean, talking about chill bump, I'm still getting them right now. It's pretty phenomenal. We were very excited about this data. Now, 100 patients, safety study, not power for outcome, so don't take it to the bank. All right, so we published that study in 2007. Unbeknownst to us, another trial was done about probably in the very similar time range, but was published in 2008. We were blindsided. Harriet, you remember this? It was like, where'd this come from? Who's doing this trial? And so a group of Chinese had done it, and they actually found almost identical type of results. I think their mortality difference was about a 48% or 47%. Their study was designed a little different. They had IM. There was 159 patients. Theirs was actually a phase 3 type of trial, but only in 1 center. So that was really the only downfall to this trial. But the beauty of it was we did 1-month outcomes for ProTECT because it was safety. We weren't funded to do any further outcomes, but these guys did both 3 and 6 months and found an improvement in the Glasgow outcome scale at both time points, which really was very supportive and very exciting. So that led us to the ProTECTIII. We submitted it to the NIH in 2008, somewhere thereabout, and they agreed that the evidence was overwhelming despite lots of I'm sure consternation that they didn't really want to believe it. And so we were funded to conduct the trial. As mentioned, I'm the PI. Our project manager is currently Bethany Lane, but it used to be Harriet over there. Raise your hand. For the first 3 years, she got it going. Very interesting, this study is running through something called the Neurologic Emergencies Treatment Trials Network, which is an NIH-funded network. The network's coordinating center is in Michigan, ran by a gentleman by the name of Bill Barson. So this is the coordinating center per se. And then the statistics is done at a completely separate place called the Medical University of South Carolina, the SDMC. And then, of course, we're the mothership at Emory for running the study. So it's kind of an interesting partnership that we coordinate together, but it's kind of nice because it keeps all of the different conflicts of interest and everything separated, because we have all of these different players in it. So it's a nice operation. It's a phase 3 double-blind placebo-controlled trial, the gold standard. Our entry criteria are this: blunt traumatic brain injury, Glasgow Coma scale of 4 to 12. Our goal is to get the drug within 4 hours. Actually, our goal is 2 hours, and we allow patients within 4. Now, I'll tell you, folks said we couldn't do it. And I'm going to show you how well we do it, okay? It's pretty amazing. Age greater than 18 is our criteria because it is a hormone. We didn't know what it was going to do to young folks, although I'll tell you that PCARN is entertaining doing a pediatric trial now. But in any case, our inclusion are, as I said, the moderate to severe, age greater than 18, blunt traumatic brain injury, and then the infusion within 4 hours. We do have a number of as all clinical trials have, a number of exclusion criteria, and you can see them here. I won't belabor them, but we can go over them if anybody's interested. But it's a powder. We compound it locally at Emory in our investigative drug center, which basically just means we dissolve it in ethanol. It's about the only thing it'll dissolve in, as it's very lipid soluble but terrible in water. And so the placebo is just the alcohol. Now it's less than 1 shot of alcohol in a 24-hour period. So if you're thinking, well, there's alcohol in there, it's a tiny amount of alcohol. We can get 50 milligrams in a cc, so it's a tiny amount that we need. And then it's mixed, because of the alcohol, it's mixed in an intralipid just before administration at the local site, wherever it is. We do a bolus dose of 14.3 cc's for an hour, and then we set it off to 10 cc's an hour for 71 hours, and then we do a Q8-hour taper over 24 hours. So that's how it's administered. It's continuous infusion over that period of time. Just sort of the basic fundamentals of any clinical trial. We, once we enroll them, we're seeking consent, but we run under something called exception from informed consent. I'd be delighted to talk to anybody about that afterwards. It's very complex, but it has been wildly, highly accepted by all the subjects that we've had. We've had, out of 755 subjects, we've had 9 whose family have withdrawn them from it. And that's basically the prevailing reason from all of those folks is he just didn't want to be a part of research. So it's one of those where you couldn't have won no matter what, because they just don't believe in research. So nonetheless, that's where we are. I'll actually show you a couple of slides on that. And we monitor AEs and SAEs throughout the process, and then our primary outcomes at 6 months, which is the Glasgow Outcome Scale, and I'll show you that. So our primary outcome is a sliding dichotomy based on their index GCS or their index motor, and this is at 6 months we measure this. We also are doing a full neuropsychological battery on those who can perform it, those with a GOAT. Anybody know what a GOAT score is? It's not that kind of GOAT. It's a Galveston Orientation Amnesia Test. If they score above a certain amount, it says they can perform these tests, and then we'll do it. So we'll be looking at all of that data. We also, in this trial, have a very strict management criteria that we push out to the sites. We formed a committee called the Clinical Standardization Team, and it is headed by Jeff Manley, a neurosurgeon at UCSF, but it has a whole variety of other neurosurgeons on it, and trauma surgeons and neurologists, neurointensivists. It's a very highly regarded folks on this committee, and we look at the Brain Trauma Foundation guidelines. Where there was evidence, we created that in our guidelines. Where there's not evidence, we developed a consensus across the sites, and so that all the sites who participate in ProTECT have to follow these guidelines as best as possible. If not, what do? We get on them. We call them, we email them, we say, you're not following the guidelines, what the hell? So we work on it as much as we can. Of course, it's not perfect, but we do the best we can. It's a very complex study. It involves a whole lot of people, and it's a team approach. This is not an emergency medicine, not a neurosurgery, not a trauma study. It's all of the above. So that is ProTECT. We are also doing a biomarker study, and we're looking at a variety of things, the S100 beta, which everybody looks at, GFAP, this UCH1, L1, and then the fibrin split products, P150. This is being done, Mike Frankel is the PI of the BioProtect, started after the original study, but he's heading this up. Interesting, we're saving blood also for new markers that are coming along, so if you have any ideas for new markers, we are storing blood for that purpose. Study update, well, when I turned this in, because you guys made me turn it in last week, which I don't like, we're now at 755 as of today, so I can give you that update, which is very nice. But at this time it was 752, 442 in BioProtect. As I said, we started later. So far we've had no serious safety concerns. We've met all our milestones. We've completed our first interim analysis, and basically after that, they told us keep going, and not only keep going, we don't want to see you again until the next interim analysis. I think that means that it's a pretty safe drug, right? So that's where we are. Our next interim analysis and DSMB meeting are in the fall. We'll be at the Neurocritical Care Society meeting in October. This is our enrollment chart. We are very close to meeting what we had hoped to meet on our enrollment goal. Had a little slow in the December-January period, but we're now picking up. I don't mean for you guys to be able to read this per se, but just to show you all the different sites that are involved in our study. Very small compared to Synapse, which has how many thousand sites? I'm just kidding. Anyway, a lot of sites. All right, so I've got 6 slides maybe just to give you a true update of the data. Now, I will, the caveat to this data is this was what we presented to the DSMB. Our statistical center will not let us pull data out and just give it. They have to have frozen the database and cleaned it before they give it to us. So this is the DSMB data. But nonetheless, it's pretty cool. 621 patients at this point in time. So a couple of things just to point out. Actually 12. I said 9, didn't I? So 12 withdrawn at this time. That's a menial amount out of the 621. And there we go. We've only lost, at this time, we'd only lost 4 to follow-up. I think it's 9 lost to follow-up, which is what we have now. But that's amazing, I think. In a traumatic brain injury trial, only losing 4 to follow-up is pretty phenomenal. Our death rate is 16.9% in this overall in the trial. Things that wouldn't surprise you were mostly male, 73%. And then we have a very nice mix of ethnicities and races. This is what the slide I wanted to show you that gives you, actually, this is the age. So we have a mean age of 39.61. And most of our patients are in this 6 to 8 moderate severe group. As you would expect, mechanism for a major trauma center is motor vehicle collision. That's predominant. So this is a slide that I'm really proud of that I want to show you, which is our time to randomization, which is 2 hours and 52 minutes. It's phenomenal. Remember, these people are injured outside the hospital, not inside. So being able to get them randomized in 2 hours and 52 minutes is phenomenal. And that's where we are. Time to infusion. The infusion has to be mixed up in the pharmacy and then delivered, and then you have problems with IVs and the patient's going to the OR and all sorts of other things. So it's a little bit delayed, but still 3 hours and 36 minutes for starting the infusion is remarkable. And so that's it. As I said, this is an EFIC study. So 65%, 66% are under EFIC, 34% under consent. So the reason I say that is that we allow the first hour to find the family to get consent. If they're not there in that hour, then we do EFIC. And so that's why you'll see these with consent. We eventually get consent from, you saw that from all the families. The discharge disposition, just to show you, most of these are going to acute rehab facilities. And then it's a little, I have to, I don't remember, I knew at the time why this number is different than our other mortality number, but safety, just so you know, there's a couple of things I wanted to show you here, is that pulmonary embolism in our cohort is only 2.7%, which if you read the literature on critical care traumatic brain injury patients, that's lower than what's out in the normal number of folks who have PEs. And DVT is similar, only 8.7% DVTs. So we are not really concerned about the coagulopathic, quote-unquote, stuff that is blamed on progesterone. Pneumonia is very common. Dave can speak to that, and even in our cohort. So we're on track with our enrollment. We're probably going to finish spring of 2014. BioProtect, the biomarker study, is fully integrated. We've got pretty good compliance with our treatment protocol. So far, no safety issues. We've completed our first interim analysis and told to keep going. And as I said, our next DSMB meeting and our next interim analysis is due out in October of 2013.